Hello friends, I am Dr. Nilesh Chandra and we will be discussing the questions from NEET exam 2020, the questions which landed up in the biochemistry section. So, we will take a quick look at the questions that were probably there, the possible options which were there and which one would be the best option to mark in the given questions. So, let us take a quick look at the questions and their answers. So, first few questions from the acid base balance, starting with this question. The lab report of a patient given below, pH is 7.2, the bicarbonate is 10 milliequivalent per liter and the PCO2 is 30 millimeter of mercury. And the question says this exemplifies which of the following disorder. Your options are respiratory acidosis, alkalosis, metabolic acidosis and metabolic alkalosis. Now very frequently we get the question of this sort in your exam. So what you have to keep in mind, generally in these questions you will get three values. You will get the pH. Obviously the pH will help us to determine whether we are looking at acidosis or we are looking at alkalosis. After pH you will get two more values. First, the bicarbonate value and second, the PCO2 value. Now here you should know the approximate normal values for them, and for bicarbonate it would be typically in the range of 22 to 28 milliequivalent per liter. For one value we can go with 24 milliequivalent per liter. Similarly, for PCO2, the range would be 35 to 45 millimeter of Hg, but if you want one value, we can go with the 40 millimeter of Hg. This should be the normal value. If it is increased, obviously value will be more than the given normal values, and accordingly it will be decreased. Now, what does the bicarbonate denote and what does PCO2 denote? See, the PCO2 will give rise to the H plus concentration. Alright, so if there is increase in PCO2, there will be corresponding increase in H plus concentration, and the bicarbonate will neutralize the H plus. So if you have more bicarbonate, you will have less H plus. Alright, and we know that H plus will determine the pH. More is the H plus, less will be the pH, means more H plus means acidosis, less H plus means alkalosis. Now keeping these 3 points in mind, we can quickly determine what we are going to find. Now pH normal value, you again know that it is in the range of 7.35 to 7.45, and we normally say it is 7.4. The mean pH value of the blood would be 7.4. So first you look at pH. Normal value should be 7.4, here we get 7.2. This means we are looking at acidosis. It is acidic in nature, so we are looking at acidosis. Now, acidosis means the H plus concentration should be increased. The increase in H plus can occur in two ways. Either there is increase in PCO2, right, or there is decrease in the bicarbonate, because the bicarbonate and H plus have opposite relationship, inverse relationship. So if the bicarbonate will decrease, H plus will increase. The PCO2 and H plus we said have a direct relationship. The PCO2 will rise, the H plus will also rise. So these are two possibilities. Now you look at which of the following changes has occurred in the question. PCO2 increase, has it occurred? Let's see. PCO2 has not increased, it has decreased. So this would be not the cause for increase in H plus. What about bicarbonate? The bicarbonate, has it decreased or increased? It should decrease to increase H plus, and here we find that it has decreased. So this should be the primary cause, the acidosis, and because there is drop in bicarbonate level, it should be the metabolic acidosis. It should be the metabolic acidosis. So like I said, the metabolic acidosis. Quickly we can put it in a graph form to recall it better. We look at the pH value. It can either be less than normal or it can be more than normal. If it is normal, we don't have to do anything, but if it is less than normal or more than normal, then we have to determine whether the acidosis or alkalosis is there and what type of acidosis or alkalosis is there. Second part would be compensation, which I will mention very briefly. So in first scenario, if the pH is less than 7.4, we say it is acidosis. Two possibilities: either it can be metabolic in nature or it can be respiratory. Metabolic means the bicarbonate value will change, and respiratory means the PCO2 value will change. So bicarbonate value is less than 24, then means we are looking at a metabolic cause, and simultaneously if there is the decrease in PCO2 value also, it is to compensate for the acidosis. So if both bicarbonate and PCO2 decrease, this means the cause is metabolic and compensation is provided by the respiratory mechanism. Opposite scenario, if the PCO2 is increased, then we are looking at a respiratory cause, and simultaneously if the bicarbonate is also increased, it is the compensation component. So these are the two scenarios, metabolic and respiratory acidosis. They may or may not have the corresponding compensation to correct for the acid base disturbance. Similarly, if the pH value is more than 7.4, then we are looking at alkalosis. If bicarbonate value has increased, it is the primary cause, we are looking at metabolic acidosis. And if the PCO2 has also increased, in that case it is compensation. If there is no change in PCO2, it is in the normal value, no compensation. If it has increased, then only we will say that compensation is there. And if the PCO2 has decreased, the primary cause will be respiratory. And if there is change in the bicarbonate, that is there is decrease in bicarbonate, then it will constitute the compensation. So using this graph, we can quickly determine what type of acid base balance is there and whether or not any compensation is occurring. Okay, so this helps us to quickly determine the acid base disorders. Very frequently in the clinical scenarios, we don't need to work this out, because most of the time we can have a handbook. You can have any handbook which has this graph called the Devonport. We have a graph called the Devonport diagram, and it just put in the values for the parameters: the bicarbonate value, the blood pH, and the PCO2 value. Put in the three values and you can quickly read out from this graph the type of acid base disturbance. Alright, so these are few points which you have to keep in mind. Sometimes, rarely, you may be asked about the difference in anion and cation, which is known as anion gap, but that is a different scenario altogether. Okay, moving on to second question, again from the acid base disturbance. In this case, what is given? Clinical presentation: a 55-year-old male presents with tachypnea and mental confusion. Okay, blood glucose level is 350 milligram per deciliter, so blood glucose level is high. In addition, the pH is 7. pH is 7 means we are looking at a type of acidosis. Now in diabetic patients, is there any type of acidosis? Yes, we have what is known as diabetic ketoacidosis. If it were diabetic ketoacidosis, the ketone bodies will be formed and they will appear in urine. So in next case, it is given urine shows the presence of ketone bodies. So we are looking at diabetic ketoacidosis, and this diabetic ketoacidosis is a type of metabolic disorder, because there is defect in the metabolism, because of which ketone bodies are rising, and it is therefore metabolic acidosis. So this is a straightforward question, which even in which you don't need to go into the values of the bicarbonate and PCO2. Simply by looking at the clinical scenario, we can depict what is the primary cause for the acidosis. Okay, moving on to next question. Now we are coming to some questions from carbohydrates. The question was, among these insoluble dietary fiber is, first, what is dietary fiber? Dietary fiber, polysaccharide in the diet which is not metabolized, right? So polysaccharide in diet which is not metabolized, all of the options are dietary fibers, but the question says insoluble among them, which is the insoluble. Now here there is some confusion because two of them are insoluble, lignin as well as hemicellulose, both are somewhat insoluble, but still the better answer would be lignin. Lignin is the better answer if you have to choose one, that lignin is the insoluble dietary fiber because it doesn't go any significant change, it cannot be digested, cannot be broken down by the anaerobic bacteria also in the gut. So lignin would be the preferred answer among the two. If lignin is not there, in that case we can choose the hemicellulose also. Okay. Another question from the carbohydrate chemistry, the proteoglycan present in a glomerular basement membrane, and various options are there: keratin sulfate 1, 2, heparin sulfate and chondroitin sulfate. Now this is one topic I always say that it is likely to land up in your exam, location of the different proteoglycans or the glycosaminoglycans. The glycosaminoglycan combine with proteins and the result is the proteoglycan. So whether you talk about the location of the glycosaminoglycan or the proteoglycan, it is somewhat similar. Let's take a quick look at the small table showing the presence of the different glycosaminoglycan in the specific tissues. In this case, for your reference, the answer is heparin sulfate. Heparin sulfate is found in the glomerular basement membrane. So hyaluronic acid, the most common glycosaminoglycan, it is present in the synovial fluid, the vitreous humor and the loose connective tissue. Most widespread glycosaminoglycan is the hyaluronic acid. Please note, glycosaminoglycan are also known as mucopolysaccharides. They are also known as the mucopolysaccharides. Then we come to the chondroitin sulfate. Chondroitin sulfate is present in the cartilage, bone and in the cornea. Keratin sulfate 1 is also present in cornea just like chondroitin sulfate, and keratin sulfate 2 present in the loose connective tissue. Alright. Heparin released by the mast cells, and heparin sulfate is present in the skin fibroblast, the aortic wall and the glomerular basement membrane. Dermatan sulfate, again it has wide distribution, commonly present in the skin and the other connective tissue. So dermatan sulfate is also having the wide distribution. So these are the 7 important common glycosaminoglycan that you have to know about, and 1 or 2 tissues where they are commonly present in significant amounts. Okay. Moving on to another question from the carbohydrates, the insulin glucagon ratio is decreased, which of the following enzyme will be most active? Now this is a tricky question. Very frequently we get the question on the insulin glucagon ratio, the action of insulin, the action of glucagon, what will happen in fasting state, what will happen in the fed state, what is happening in which enzyme is activated in phosphorylated form, which enzyme is activated in the dephosphorylated form. Like that question will keep coming again and again. There is a very simple mechanism by which we can solve these questions. I will quickly mention that how to handle them. The insulin glucagon ratio, if you write like this, insulin glucagon ratio. Now we know that the insulin and glucagon have a reciprocal relationship. What we mean by reciprocal relationship? Insulin increases, glucagon will fall. If insulin falls, glucagon will increase. Alright. Now, if insulin increases, the glucagon will decrease. Reciprocal relationship. So, what is happening? Numerator has increased, denominator has decreased. What will happen to the net value? The net value will also increase. More numerator, less denominator, the net value will increase. Let's look at another scenario. Insulin decreases. What will happen to glucagon? Glucagon will increase. Reciprocal relationship. So what happened? Numerator has decreased, denominator has increased. What will happen to the net value? The net value will become less. Now, very interesting observation if you look carefully. If insulin increases, the insulin glucagon ratio also increases. If insulin decreases, the insulin glucagon ratio also decreases. So when you look at a question telling you insulin glucagon ratio, for the purpose of the question, don't look at the glucagon ratio, just read it as insulin is increased. Ratio will only increase when insulin will increase. Alright. So now we should only know the action of insulin. If you know the action of insulin, you will be able to tell the questions for the insulin glucagon ratio. Second point, when will insulin rise? When there is more glucose, means fed state. When will insulin fall? When there is low glucose, that means you are looking at fasting state. So when I say fasting, it means low insulin. When I say fed state, it means more insulin. And if you know the action of insulin, then you can quickly tell us what is going to happen. Alright. So again, the second, fasting and fed, is also converted to action of insulin. Third, insulin mostly causes dephosphorylation. Remember this point, insulin mostly causes dephosphorylation. And now we come to the actions of insulin on the different pathways, after which the picture will become very clear. Okay, for this case the answer is glucose 6 phosphatase. We'll come back to that. Action of insulin, it increases glycolysis, it increases glycogenesis, it increases lipogenesis, it increases HMP shunt, it increases amino acid synthesis and cholesterol synthesis. So effectively what it does, it increases glucose consumption. And this should be obvious. Why? Because insulin increases only when there is increase in the glucose. So action of insulin is to decrease the glucose by consumption. At the same time, insulin also decreases the glycogenolysis, it decreases the lipolysis, it decreases the gluconeogenesis, and it decreases the ketogenesis, meaning it decreases new fuel formation, and the fuel are glucose, the ketone bodies, and the fatty acid. So any mechanism by which these will become available is prevented. It is prevented by the action of insulin. Now what we said, the insulin mostly causes dephosphorylation, meaning the enzymes of these pathway are active in dephosphorylated form, and similarly the enzymes here are inactive. They are inactive in dephosphorylated form. This will cover most of the enzymes related to metabolic pathway. When you look at enzyme, you can quickly ascribe it to a particular metabolic pathway, and by remembering this table, you can quickly determine what will happen to enzyme activity in a phosphorylated state and what will happen to enzyme activity in the dephosphorylated state. This is a simplified version to remember the activity of most of the enzymes, the metabolic pathways, when they will be activated, when they will be inactivated, what happens when there is change in the insulin glucagon ratio, what happens in the fed state, what happens in the fasting state. So looking at the previous question, if you go back, you will find the question says insulin glucagon ratio is decreased. This means the insulin will decrease. This means whichever pathways are normally decreased by insulin will now be activated, and whichever pathways are normally activated by insulin will now be decreased. Glucokinase, part of the glycolysis or the glycogen synthesis, which is normally activated by insulin, so will be decreased. Hexokinase, again same, decreased. Phosphofructokinase, again for glycolysis, it will be decreased. Glucose 6 phosphatase belongs to the gluconeogenesis, and we said new fuel formation is inhibited by insulin. If there is less insulin, new fuel formation will increase, and that is why the glucose 6 phosphatase will be activated when insulin glucagon ratio is less. Okay, moving on to next question. Now this question is from the lipid section. In a preterm baby with respiratory distress syndrome, which of the following lipid would be deficient? Now all of you know the answer to this question is lecithin. To be more precise, it is dipalmitoyl lecithin. But what you have to know is lecithin, the more correct name for lecithin is the phosphatidylcholine. So the correct answer is dipalmitoyl phosphatidylcholine. There was another question in the exam which is asking about the type of cell which is not mature. So you should remember the phosphatidylcholine is being produced by the pneumocytes type 2. So in the preterm baby, these type of cells are not mature, and that is why the dipalmitoyl lecithin formation is not sufficient, giving rise to the respiratory distress syndrome. Both the questions that come, what is less and which cell is not mature. The pneumocyte 2 question belongs to the physiology section, so I have not taken that up. Okay, another question, and this is a very important question because the metabolic syndrome is currently a hot topic. The NCEP-ATP3 criteria is for metabolic syndrome, and you should know the parameters which are included in the metabolic syndrome criteria. So the question says, which of the following is not included in metabolic syndrome? And the options are hypertriglyceridemia, high LDL, central obesity, and hypertension. Please note, in this case the answer happens to be high LDL. This is very important to remember that in the definition of metabolic syndrome, LDL is not included. LDL is not a part of the definition of metabolic syndrome. So what is a part of metabolic syndrome? Let's take a quick look. As per NCEP-ATP3 definition, metabolic syndrome is present if 3 or more of the following criteria are met. Out of these, one is essential. We will start only if this criteria is there, and that is waist circumference more than 40 inches or 90 centimeter in males and more than 35 inches or 80 centimeter in the female. This is essential. Only if this is there, then we can apply the other criteria. If it is not there, we cannot apply the other criteria. We will not be talking about the metabolic syndrome. Now, 2 or more among 4 different criteria should be there. Blood pressure, systolic more than equal to 135 millimeter of mercury, diastolic more than equal to 85 millimeter of mercury. The fasting triglyceride more than equal to 150 milligram per deciliter. The fasting HDL in case of female less than 50 milligram per deciliter. In case of male, the value is less, that is 40 milligram per deciliter. And lastly, the fasting blood sugar more than equal to 100 milligram per deciliter. Okay, so essential criteria and 2 or more of the remaining 4, if they are present, then we call it the metabolic syndrome. Metabolic syndrome has been associated with a large number of diseases, and that is why this definition is very important. You must know about the criteria which is included in the metabolic syndrome. It is likely to be asked again and again and now. Now we move on to the enzyme section. True about non-competitive inhibition is, now here we are talking about the change in enzyme activity due to different type of inhibitors. Very briefly, I will mention the type of inhibitors. The inhibitors can be 2 broad types. They can be reversible and they can be irreversible. Please note, mostly in your exam the question will focus on the reversible type, because it is only the reversible type where we can have some pharmacological management to remove the action of inhibitor. In irreversible type, only symptomatic management is there, so clinically it is not very significant. Most of the questions will focus on the reversible type of inhibitors. The reversible type of inhibitors will be of 3 subtypes: competitive, non-competitive and uncompetitive. Please note, non-competitive is a special subtype of a category called the mixed inhibitor, but the exam will only ask about the non-competitive type. I'll quickly give you the important points about the three. First, what happens to Km? In case of competitive, the Km increases. Alright. In B, the Km remains the same in non-competitive, and in uncompetitive the Km decreases. I am sure you know what is Km. Km denotes the affinity between the enzyme and the substrate. Km and affinity have a reciprocal relationship, means more affinity less Km, less affinity more Km. Then we have the value of Vmax. In case of competitive inhibition, the Vmax will remain the same. In case of non-competitive and in uncompetitive inhibition, the Vmax will decrease. Last point to keep in mind is the point of attachment, where the inhibitor will go and attach on the enzyme. In case of competitive inhibition, it attaches at the active site. In case of non-competitive inhibition, it attaches at the allosteric site. But most important and different is the point of attachment for uncompetitive inhibition, where the inhibitor attaches only to the enzyme substrate complex. So these are the 3 points which you have to keep in mind: the change in Km, the change in Vmax, and the point of attachment of the inhibitor in case of the 3 different types of the reversible inhibition. They are the competitive, non-competitive and uncompetitive. These are the 3 different types of inhibitors under the reversible inhibitor category. You have to know the changes. Now when we say non-competitive inhibition, what happens? The Km will remain the same and the Vmax will decrease. So the correct answer should be A. The Vmax remains the same, sorry, Vmax decreases and the Km remains the same. Okay. Moving on to next question. Replacing alanine by which amino acid will increase UV absorption of protein? Now we are moving into the protein section, amino acid and protein section. The options are tryptophan, leucine, proline and arginine. Now what we have to understand here, the absorption at 280 nanometer or in the UV range occurs by aromatic compounds. So here you should know which are the aromatic amino acids. The aromatic amino acids are tyrosine, phenylalanine, histidine and tryptophan. So if you know the aromatic amino acids, immediately you can give the answer that absorption at 280 nanometer will increase only when we add an aromatic amino acid. Among the given option, the only aromatic amino acid is the tryptophan. So the correct answer should be tryptophan. Tryptophan, if replaces any other amino acid, the resulting protein will have increased absorption at the 280 nanometer in the UV range. Then we have question asking about the synthesis of one of these special products, the nitric oxide. Nitric oxide is synthesized from which of the amino acids? Now frequently you will get questions talking about special products and the source from which they are coming. In this case, nitric oxide is synthesized from arginine by the enzyme nitric oxide synthase. We know that nitric oxide is a second messenger. It helps in the relaxation of the various muscles, particularly in the blood vessels, and it is a very important second messenger. Let's take a look at some of the other important special products and from where, which amino acid are they coming. The tyrosine gives rise to multiple special products. First and foremost, the catecholamine. in the adrenal medulla. The catecholamine meaning the dopamine, norepinephrine, epinephrine come from the tyrosine. In the thyroid gland, they give rise to the thyroxine hormones, the T3 and T4. So, tyrosine gives rise to the catecholamines, it gives rise to the thyroxine hormones, and in the melanocytes, it gives rise to the melanin. It gives rise to the melanin. First dopa is formed and then it is converted to melanin. So, tyrosine gives rise to the catecholamines, to the thyroxine hormones and the melanin. Then you come to tryptophan. Tryptophan gives rise to melatonin, serotonin and niacin. Niacin meaning vitamin B3. Please remember 60 milligram of tryptophan is consumed to generate 1 milligram of niacin. 60 milligram tryptophan will give rise to 1 milligram of niacin. Then from arginine we just said nitric oxide is obtained. From histidine and from glutamate we get the corresponding molecules histamine and GABA. Both are produced by the decarboxylation of the parent amino acid. By decarboxylation of histidine you get the histamine. By decarboxylation of glutamate you get the GABA. GABA means the gamma amino butyric acid. The gamma amino butyric acid, GABA, obtained from glutamate. So, these are some of the very important special products obtained from the different amino acids which you should know. Okay. Now, we move on to some of the very important clinical questions which were there in the exam, long questions. All of you know that long clinical questions were there in the NEET exam this year. Okay, first question, 4-year-old boy of a first degree consanguineous couple. So, when we say consanguineous couple, what we are trying to emphasize is there will be likelihood of increased incidence of autosomal recessive disorders, more likelihood of autosomal recessive disorders. It was noted by parents to have darkening of urine to an almost black color when it was left standing. So, blackening of urine on standing, normal siblings and no other associated medical problems. Growth and development to date are normal. Now, this is a very special scenario where although there is blackening of urine in the child, there is no associated clinical signs symptoms until the 3rd and 4th decade of life. At that time there are various clinical presentations, but before that there is no other clinical presentation except blackening of urine on standing. The condition is known as alkaptonuria. The condition is known as alkaptonuria and the enzyme defect in alkaptonuria is homogentisate oxidase. The enzyme defect is homogentisate oxidase. In the 3rd and 4th decade they will present with another set of problems. They will have back pain. On examination you will find that there is stiffness of the back. They will have early onset erosive arthritis. Early onset erosive arthritis, because the homogentic acid crystals will be there in the synovial fluid. Whenever there is movement on the joints, the crystal will rub the synovial membrane, resulting in erosive arthritis and what is known as ochronosis. This is blackening of the skin over various parts of the body. This is known as ochronosis. This is again because the deposited homogentic acid gradually gets oxidized over years and decades. In the atmosphere, partial pressure of oxygen is high, so quick oxidation. In the body, partial pressure of oxygen is low, so oxidation takes a very long time. So the blackening occurs over decades. So ochronosis is the blackening of the deposited homogentic acid. All of this will appear in 3rd and 4th decade. Before that there are no other signs symptoms except darkening of urine on standing in case of alkaptonuria. Alkaptonuria belongs to a group of cluster of disorders in the phenylalanine tyrosine metabolism complex. So, I will quickly enumerate the different disorders in the phenylalanine tyrosine complex without going into the details, just for a recall. The phenylalanine normally metabolized to tyrosine by the enzyme phenylalanine hydroxylase. When the enzyme phenylalanine hydroxylase is deficient, we get the classical type of phenylketonuria, also known as the phenylketonuria type 1. For this reaction, we require the coenzyme cofactors, the tetrahydrobiopterin, which is reduced to dihydrobiopterin and then recycled to the tetrahydrobiopterin. Sometimes the defect is in the cofactor part, and this is also known as phenylketonuria but malignant type, which is further subdivided into 4 type 2, 3, 4 and 5 depending on the type of enzyme which is involved, not going into the details. Tyrosine is then metabolized to the hydroxyphenyl pyruvate by the enzyme, the tyrosine aminotransferase. Deficiency of the enzyme is known as tyrosinemia type 2, also known as the Richner-Hanhart syndrome. The Richner-Hanhart syndrome. The hydroxyphenyl pyruvate is then converted to the homogentisate by the enzyme hydroxyphenyl pyruvate oxidase. Now here there can be two type of disorders. One, when the enzyme is not there. When the enzyme is not there, there is deficiency of the enzyme. The neonatal tyrosinemia, also known as tyrosinemia type 3, it is very mild. Most of the patients are asymptomatic. Rarely you can have some amount of mental retardation. Sometimes the enzyme is defective. When the enzyme is defective, the metabolism stops in between and the final product is not formed, and in that case we have the defective enzyme is there. The homogentisate is then converted to the fumaryl acetoacetate, and this was the enzyme that we are talking about, the homogentisate oxidase. The condition we said is known as the L-captonuria. Finally, the fumaryl acetoacetate is converted to fumarate and acetoacetate. The enzyme which is used here is fumaryl acetoacetate hydrolase. Deficiency gives rise to the tyrosinemia type 1, which is the most severe type of tyrosinemia, also known as tyrosinosis. Right? And here what happens, the liver and kidney are affected, so it is also known as the hepatorenal tyrosinemia. The defect in the enzyme gives rise to a intermediate called Hawkinson, and the condition is known as Hawkinsinuria. Normally it is not mentioned in the list of the phenylalanine tyrosine complex because it is not due to deficiency of the enzyme, rather it is due to a defective enzyme. So remember, the Hawkinsinuria is also part of the phenylalanine tyrosine complex. However, the cause is not a deficiency or absence of enzyme, rather it is due to a defective enzyme. Okay. Moving on to next question, again about the proteins. True about protein structure. Which of the following is true about the protein structure? And these were the 4 statements which are provided. Secondary structure is decided by amino acid sequence. Now we know that this is false. Amino acid sequence will only determine the primary structure. Tertiary structure is responsible for stability. This appears to be correct, but let's look at the other options. Sulfide bond is present in the secondary structure of protein. In secondary structure of protein, we only have the non-covalent bonds, and obviously sulfide bond is a covalent bond, so it cannot be a part of the secondary structure. So sulfide bond will be there in the tertiary or the quaternary structure, not in the secondary structure. Primary structure is lost on heating or denaturation is also incorrect. On heating only the secondary, tertiary and the quaternary structure is lost, we know this. So this statement is also correct, not correct. So the correct statement is tertiary structure is responsible for stability, which is the answer for this question. Next question. Bilirubin in serum can be measured by. Now I have been saying this again and again, certain analytes and the related tests are likely to land up in your exam, bilirubin being one of them. So bilirubin in serum is detected by the Vandenberg test. What about the other options which are given? Ehrlich reaction, also known as the Ehrlich aldehyde test, can be used to identify urobilinogen or the porphobilinogen, right? The urobilinogen or the porphobilinogen. The Schlesinger reaction is used to identify urobilin. The Fauchet's reaction is used to identify the bile pigments. meaning for the bilirubin. These three tests, the Ehrlich reaction, the Schlesinger reaction, the Fauchett's reaction, is commonly used for the urine sample. The Vandenberg test is commonly used for the serum sample. Alright. When we say urine sample, most of these tests happen to be qualitative only. What do you mean by qualitative? They will tell us whether a substance is present or not. They will not tell us the quantity, they will only tell us whether the substance is present or not. So, these three tests in the urine are qualitative in nature. The Vandenberg reaction is a test for serum, and most of the serum reactions are quantitative in nature. Most of the serum reactions are quantitative in nature. They will tell us the exact amount because we are also running standards, and then we do the colorimetric estimation of the concentrations. Okay. Moving on to next question. Now we are moving to the molecular biology. The amount of thymine in a given sample of DNA is 28%. What will be the amount of the guanine in the same sample of DNA? Now this is a very simple straightforward question based on what is known as chargaffs rule. All of you know this. The chargaffs rule has very simple 2 points. The adenine combines with thymine, the guanine combines with cytosine. That is why the amount of adenine will be equal to amount of thymine, the amount of guanine is equal to amount of cytosine. Now question says the amount of thymine is 28%. This means the amount of adenine is also 28%, right? Let us say the amount of the guanine is X, in that case amount of cytosine will also be X, and when you add all of them, total should be 100%. So 100% should be equal to A plus T plus G plus C. This means it should be equal to 56 plus 2x, 56 plus 2x. In other words, 2x is equal to 100 minus 56, which is 44, or x should be 22%. So by simple elementary calculation, the answer should be 22%. The answer should be 22%. The question is based on the very basic rule which is known as the Chargaff rule. Chargaff rule tells us adenine equals thymine, guanine equals cytosine in double stranded DNA, which is given in the question, right? Okay, let's move on. Again, question from the molecular biology. Differential expression of B48 in the intestinal epithelial cells is due to which type of modification? Now various modifications which can occur in terms of expression can be the modification of DNA, there can be the RNA splicing, which is part of the post transcriptional modification in the nucleus. There can be changes in the RNA in the cytoplasm, which is called the RNA editing, and sometimes there can be interference by the micro and the silencing RNA. Alright, so these are the various options which can occur. The RNA interference by micro and silencing RNA. The RNA splicing is a part of post transcriptional modification occurring inside the nucleus, right? The DNA processing also occurring in the nucleus, and RNA editing occurring in the cytoplasm. Now if you look at the messenger RNA in case of epithelial cells as well as liver cells, once it is released from the nucleus, we have the same messenger RNA for both the cells, meaning the splicing and the DNA processing which is occurring in the two tissues is the same, there is no difference. Now we are left with RNA interference and RNA editing. Now RNA interference, the action of RNA interference is decrease in amount of protein. It does not change the type of protein, it only decreases the amount of protein which will be formed. So again, this cannot be the cause for B48 being formed from an RNA which is going to give us the B100. So we are left with RNA editing. RNA editing occurs in case of epithelial intestinal epithelial cells, whereby if we have this messenger RNA at about 48% of the length, a stop codon is generated due to RNA editing. So when the translation occurs, only this part is translated. A smaller protein is formed due to translation of only 48% of the messenger RNA, called the B48 apolipoprotein. If all of the RNA is translated, we get the B100 protein. If only 48% is translated, we get the B48 protein. Remember, 48 doesn't denote the mass, it denotes what percentage of RNA has been translated. Okay, moving on. Which of the following is the etiology of Werner syndrome? Now, Werner syndrome is one of the types of early aging syndrome. Now, this is one of those questions which I say you need not know the answer, because these questions will come rarely. Sometimes always one question, two question, five questions are there which is beyond the scope of our discussion. So even if you don't know, it doesn't matter much, but just give you the answer. Werner syndrome is a type of early aging syndrome where there is defects in the DNA helicase gene. As a result, we get the defective DNA helicase and early aging will occur. So answer in this case will be DNA helicase defect. Now again, another question from molecular biology: which of the following drugs act by inhibiting DNA replication? I am sure most of you know the answer to this question. It is 6-mercaptopurine. 6-mercaptopurine. So purine and pyrimidine analogues can interfere with the DNA synthesis by getting incorporated into the DNA, but preventing the further elongation of the polynucleotide. So purine, pyrimidine analogues will be common molecules which can inhibit the DNA replication. Another question was there talking about the molecules which inhibit the transcription, that is formation of RNA from the DNA, and the various options were rifampicin, nitrofuran, ciprofloxacin, novobiocin. Now this is a somewhat uncommon question, so I'll give you the small list of molecules which are commonly used and which interfere with the transcription. In prokaryotes, some inhibitors will bind with the beta subunit of the RNA polymerase. In prokaryotes, the RNA polymerase commonly has 5 subunits: 2 copies of alpha, 2 copies of beta known as beta and beta dash, and the omega. So these molecules are binding to the beta subunit. They are binding to the beta subunit, resulting in prevention of the polymerase from attaching to the DNA. Rifampicin, streptoviracin, streptoglycidin, heparin and rifamycin. These are the drugs which bind to the beta subunit. Some molecules can also attach to the DNA and prevent the polymerase from interacting with the DNA. An example is actinomycin D, also known as dactinomycin. Now, is there any important example of molecule which inhibits transcription in the eukaryotes? Yes, the answer is alpha amanitin, obtained from the mushroom Amanita phyllets you read in pharma, and it inhibits the RNA polymerase 2 to a significant extent. It also inhibits polymerase 1 and 3, but their inhibition is less. The most prominent inhibition is for RNA polymerase 2, which is the more important type of polymerase for the eukaryotes. Okay, so that's about inhibition of transcription. Now we go to the domain of vitamins. Lots of questions came from vitamins, focused on only 2 of the vitamins. So we'll quickly take a look at which all questions were there from the vitamins. First question: vitamin B12 cyanocobalamin is found in. Please note, vitamin B12 is produced only by bacteria. As a result, the B12 is found only in animal food. It is not found in the plant food. If you look at the different options, only one option is animal food, rest all plant. So it will not have vitamin B12. The answer is animal product. In plant food you can have B12, only one condition: contaminated by infection. For example, there is fungi infecting the crop. In that case, you can have some amount of vitamin B12. Okay, otherwise B12 will not be there in the plant food. Long, you can see a very long clinical question. 30-year-old woman comes with complaint of easy fatigability, exertional dyspnea and weight loss. She also complains of frequent fall. Frequent fall is commonly due to some sort of neurological deficit. Okay, the easy fatigability can have multiple causes. Let's see. Physical examination revealed that there was bilateral decrease in vibration sense. Again, we are looking at some sort of neurological deficit. Hemoglobin level was 8.2. So anemia can explain the fatigability, the exertional dyspnea and the weight loss. She was treated with folate supplementation. They must have seen the peripheral smear which would show the megaloblastic anemia, and that's why the folate supplementation was given, and the anemia thereafter improved, but the neurological symptom worsened. Now we know the megaloblastic anemia can be because of two reasons: folate deficiency and B12 deficiency, because B12 is required for regeneration of folate. If B12 is not there, we have what is known as folate trap. The folate is there but not in the active form, so it cannot be used. B12 deficiency will also result in neurological damage because it is required for some reactions in the lipid formation which help in the stabilization of the neurons. Now, if the neurological deficit is increasing on folate supplementation, we are obviously looking at B12 deficiency which is getting aggravated. So, folate therapy caused use of B12 stores aggravating the symptom should be the correct answer in this case. Folate not absorbed, if it is not absorbed, then anemia would not improve, not correct. Pyridoxine deficiency not related to the neurological deficit. Deficiency of folate reductase in the CNS unlikely to be the cause. Folate therapy caused use of B12 stores aggravating symptoms. Another question. 50-year-old man presents with paresthesia, neurological deficit, hemoglobin 6.8, anemia, peripheral smear shows macrocytosis. Macrocytosis means we are looking at megaloblastic anemia, neutrophils with hypersegmented nuclei. Endoscopy reveals atrophic gastritis. What is the role of gastritis in any of the vitamin absorption? The stomach produces the intrinsic factor which is required to attach B12 and help in absorption. So intrinsic factor will be produced by the stomach. If there is atrophic gastritis, intrinsic factor will not be produced, B12 absorption cannot occur successfully. So again, the B12 deficiency is likely to be the cause in this patient. 3 questions so far from vitamin B12. A young man met with motorbike accident that had injury to ileum and jejunum. Therefore, the entire ileum and partial jejunum was resected. Which of the following is patient most likely to suffer from? Meaning, which particular molecule is exclusively absorbed in the ileum? Again, the answer is vitamin B12. Once it attaches to the intrinsic factor, the final absorption occurs in the ileum. If whole ileum has been resected, B12 will not get absorbed and it will result in a vitamin B12 deficiency. 4 questions from B12. Remember the water soluble vitamins B1, B2, B3, B5, B6, B7, B9 and B12. These are the vitamins in the B complex. They are very frequently asked because of the associated clinical scenarios. More questions from vitamin deficiency. Female on maize as a staple diet presents with history of diarrhea and necklace pattern lesion on the neck. It is likely due to which vitamin deficiency? Now maize is rich in leucine and less amount of niacin. Both of them together result in niacin deficiency. Niacin, that is vitamin B3. Pyridoxine, please remember, is B6. Oh, this option is missing. I think it would be B9, I'm not sure, maybe B9. Okay, so here the answer is niacin. We are looking at the classical necklace pattern lesion which is known as the Casal collar or Casal necklace seen in the pellagra. Pellagra has the 3 Ds: diarrhea, dermatitis, dementia. So diarrhea is given, the dermatitis is given, dementia has not been mentioned, but that is also seen. So question 1 from niacin. Question 2, image based. The Casal collar has been shown in the diagram. This question was also there in the AIMS exam. They had given the image showing the Casal collar or the Casal necklace, and the question was asked, what is the most likely cause for this presentation? So again, the niacin deficiency, Casal collar or the Casal necklace. So I'll quickly talk about pellagra. The 3 Ds: diarrhea, dermatitis, dementia, and you can also have the glossitis. This glossitis resembles the glossitis in the A riboflavinosis. B2 deficiency also has the glossitis and stomatitis. If untreated, the 3D will merge to form a big D. This big D is death. The Casal collar or necklace, I told you, is due to the pigmentation in the C3 C4 dermatome. The C3 C4 dermatome causes a severe pigmentation, and typically historically seen in populations which consume large amount of maize and sorghum, because the niacin is not available for absorption, and the content of the amino acid content prevents the conversion of tryptophan to niacin. Whatever tryptophan is there cannot readily be converted to niacin due to high concentration of leucine. Another question from vitamin: which vitamin in supraphysiologic dose causes macular edema and macular cyst? The answer again is niacin. So these were, you saw 7 questions from water soluble vitamins, 4 from B12 and 3 from B3, that is niacin. Which of the following clotting factor in a patient on warfarin therapy would have decreased gamma carboxyglutamate residue? Now first you should ask yourself, what will happen if there is warfarin therapy? Then we'll look at the options. Warfarin therapy will inhibit the action of vitamin K. It will inhibit the action of vitamin K. Now we can look what is the action of vitamin K. Which of the following factors are undergoing carboxylation of the glutamate residue? And the answer should be 2, 7, 9, 10. These residues undergo gamma carboxylation with the help of vitamin K, and if vitamin K is not there, these factors will be affected. They will not be able to act properly because the gamma carboxylation is required for the activation of these factors. So these were the questions asked in the NEET exam. 2 points to give to understand from the NEET exam: large number of questions. We discussed, I think, 24 questions from the biochemistry section. I have left 1 or 2 questions which were overlapping with physiology and the pathology. There was one inheritance pattern shown for the autosomal dominant disorder, and then 1 more question from physiology was there which overlaps with biochemistry. I have left that outside. So large number of questions from biochemistry, 24 I have discussed. 2-3 more questions I could have included which fall in the biochemistry section, number 1. Number 2, large emphasis on the vitamin section and super large emphasis on the clinical application of biochemistry. See, someone can argue that all of these are clinical cases, but you don't need the clinical knowledge to solve these questions if you know the basics of biochemistry. So biochemistry continues to be very important to achieve a good score in your exam, simply because if you know the question, it's very easy to solve. There is no scope for confusion. The questions from biochemistry section are clear cut, either you know it or you don't know it. If you know it, very likely to score high, and a lot of students end up scoring in the range of 90 to 95% in the biochemistry section if you have done it properly. So take care, keep revising biochemistry, and best of luck. Thank you very much.